Hello everyone. We have spoken about in blood, we have spoken about the introductory part of the blood and then all the formed elements. First, we spoke about, of course, RBC or the erythrocytes. Um, Here we spoke about all of the, there's no type, basically the structure, disorders, all of that. Then we spoke about leukocytes, their structures, the types, and the disorders. And then finally, we spoke about platelets as well. And we also spoke about how these three formed elements originate and their functions, of course, each of their functions. Now we'll talk about some blood process or some function of blood, which first one is basically we'll talk about hemostasis or what is this. So how this actually is related to blood clot formation and how it is related to platelets. We'll talk about some disorders related to that, and then finally we'll talk about blood groups and some other testings related to that. So, what is hemostasis? Hemostasis is the fast series of reaction that can stop bleeding. All right. So this actually requires clotting factors because the blood stop needs to stop bleeding. So naturally, it will require to clot to become a little bit solidified so that it does not, so we don't keep losing bloods anymore. And who creates these substances? It is created by platelets. There are three steps into this. Firstly, vascular spasm. This is after you have an injury. Then the platelet plug formation, and then finally is the coagulation of the blood clotting itself. So first one, suppose there is a vessel injury, your tissue is injured, right? So the tissue will undergo some vasoconstriction so that there is a vascular spasm. Okay, so this can happen as a direct injury after the direct injury of the blood vessel. And our blood vessels in the inner side, actually it is lined, if you remember, in inner side it is lined by smooth muscles. So here after you have this injury, then you will have, then the spasm happens, and this actually gives us a little bit of pain reflex also. And then certain chemicals are starts to release from the smooth muscles endothelial cells as well as platelets. Okay, now this actually kind of the spasm little bit reduce the blood flow until the next steps kicks in. So what are these next steps? Firstly is called the platelet plug formation, the second step. So which means that the platelets stick to collagen fiber. So when our tissue, the endothelial cells are damaged, for example here is the tissue damage, then it is critically important for collagen fibers to get like exposed or to get created over here, so that this actually helps in the plug formation as well as the repair process, right? So underlying collagen fibers will be exposed, and then the platelets will start coming to that site. But initially the platelets cannot stick because the collagen fiber is not exposed. And now there are certain chemicals like nitric oxide and prostacyclin or secreted by the endothelial cells act to prevent immediately the platelet sticking so that the collagen is properly exposed. There's a special factor called von Willebrand factor that stabilizes the platelet collagen addition. So therefore now the platelets are here, and that will actually and slowly the platelets are also converted into sticky substances. Okay, why it is necessary? So the sticky, so they can stick together and actually form the plug. Okay, there is ADP that causes more platelets to stick and release their sticky content. There's some other components like thromboxane and then serotonin that actually make sure there is more spasm, and more the spasm means more platelets. That's actually a stimulus, and then more platelets will come towards the injury. Now this is a positive feedback cycle. If you remember, what is positive feedback cycle? As long as the tissue like this job is done, more and platelets will come in, will start secreting, will start coming to the side, and they will start to stick with each other. Okay, that's a and until the whole plug formation is done, this will continue to happen. Okay, but this actually happens in smaller tissue. This is fine in smaller injuries, but if you have bigger injury with like where there's a lot of blood loss is there, then we will need additional medical help. Now what happens after this? Now this is the platelets a form of a plug. Now the blood has to clot itself. Okay, so that is called as the coagulation or the blood clotting. So this cannot happen without the help of a protein called fibrin. Okay, now what is happening here? The blood itself is uh, converted into from the because blood is liquid, right? So liquid is converted into gel substance, and fibrin actually forms the protein fibrin forms a mesh that traps the RBCs, the red blood cells, and then the platelets, everything together, and then that's how the clog forms, the solidified gel kind of substance forms. Okay, and there are several clotting factors are involved in this that actually the fibrin actually regulates. For example, there are clotting factors from 1 to 13. There is vitamin K. All these are required for this coagulation factor. Okay, for this gel to form, the solid stuff to, not solid, semi-solid stuff to form. Here are all your blood factors, or these are called procoagulants or blood clotting factors. Fibrinogen is one of the main one. Then there is prothrombin, there is calcium, there is tissue factor. These four are the main one. There is fibrin, FSF, fibrin stabilizing factor. Many others are there, and they have several various kinds of sources. You have to remember the first four, actually, the tissue factor and what is their source. For example, fibrinogen is secreted from liver, prothrombin from liver, tissue factor from tissue cells, whichever tissue it is, calcium from plasma, all of that. Now the clot, after it is formed, it needs to be retracted because it cannot be there, because after the clot is a temporary process, so that this damage is like kind of blocked, that no more blood loss happens. But this is not a permanent process. Now this injured tissue needs to be repaired, so that has to happen. That's a permanent process. So therefore the clot will retract after the tissue damage is like setting in. So what happens, the platelet derived factor PDGF, it is released by these platelets. They actually stimulate the division of the smooth muscle endothelial cells inside. So here the cells that are smooth muscle cells are there, their cell division is increased, and that is stimulated by the platelet factors. So because they will rebuild the blood vessel wall, right? So that's how the blood vessel is repaired itself. Now fibrinolysis is basically by the which, and fibrinolysis and clot retraction, these are the process by which the clot is therefore basically removed. So clot retraction means that actin and myosin in the platelets contract within 30-60 minutes. This is like after the tissue repair has started. All right, now the contraction pulls on fiber strands, squeezing the serum out of the clot so that it becomes dry. What is this serum? Serum means is only the clotting factor will be there in that plug, not the plasma anymore. All right, this draws the ruptured blood vessels kind of edges together, draws them together, because the plasma is like pulled up, the serum is there, so serum is not there, sorry, and then therefore that is called as a clot is retracted, it comes close together. Okay, the whole thing comes close together. Then of course the PDGF has its own role. It helps the endothelial cells to divide, stimulates that. And then there is finally fibrinolysis, which means process by which the clot is completely removed, and this only happens after the repair is done. Okay, begins in 2 days after an injury and continues until the entire, like, fibrinolysis complete. Fibrinolysis means the fibrin protein that was there, that is completely lysed, that is like completely dissolved. Okay, and that can take time depending on the intensity of the injury. And this, when fibrinolysis is done, that means the clot is now entirely removed. It can start from 2 days, begin within 2 days of the clot formation or the plug formation, and then it will continue until the entire thing is done. Now, what are the disorders of hemostasis? Means that blood is not clotting properly, right? So this is, first of all, it is called as the thromboembolic disorder, means undesirable clot formations, like blood will be clot wherever it is not required to form like clot. The other type is that blood will not clot. This is particularly called as hemophilia or thrombocytopenia. Okay, In DIC, or disseminated intravascular coagulation, or DIC, involves both type of these disorders. It's kind of a little bit rare, but can happen. So hemophilia simply means hemophilia is not very uncommon. Hemophilia A is actually the most common hemophilia. It means that the factor 7 means the clotting factor 7 that is deficient, that is not there. So therefore the clotting of the blood becomes troublesome. So 77% of people who develop hemophilia, they actually have hemophilia A. The rarer ones are hemophilia B and C. This means prolonged type like bleeding, and sometimes some genetically engineered factors, medicines has to be given for the blood to clot. Okay, the second process is called blood transfusion. What is blood transfusion? Blood transfusion is, of course, cannot happen naturally. So if we have a significant type of blood loss, then you must have heard that when we have significant blood loss, maybe from a chronic accident or something, then we have to be given blood from outside, right? Now, body little bit can compensate for its own blood loss. If it's a small blood loss, it will compensate. Okay, so how it will reduce the volume of the affected vessels, this blood clotting that we just studied, and then setting up more and more RBC production. But if it's too fatal, for example, a big accident has happened, then we have to be given blood from outside. That is called transfusion. So why it is required? We have to restore the blood volume. So immediately we'll see that if we have too much low blood volume, then that's a shock and that death can happen. So immediately when a patient comes to the hospital with this kind of a shock, then they're given with saline. So what is saline? Is a multiple electrolytic solution or ringer solution, which has the same concentration as the plasma. So that this electrolyte actually compensates that. So it does not do anything else, but it just compensates for the volume. And then finally we need actually RBCs, because this plasma obviously is not doesn't have any RBC which will carry oxygen, and our tissues will need oxygen to perform, right, to work. So therefore then that comes whole blood transfusion. This is only happens when blood loss is very rapid and very substantial. Okay. And infusion means infusion of packed red blood cells, PRBCs, plasma, and WBC. Both, this is what is preferred, and that is given for restoring oxygen carrying capacity. Now, blood banks will typically store bloods, and human blood life is approximately 35 degrees Celsius under proper conditions. Now, before giving a blood, we have to determine blood typing. We cannot give just randomly any blood to anyone else, because human beings, we do have types of blood. Okay, so this, if we have a certain type of blood, then we need the same type of blood or similar. So now we'll talk about blood typing. So how are human blood groups determined? By the presence of antigens. So this is our blood cell, like red blood cells, we have several types of antigens sitting on these blood cells, and that determines our blood type. What is our blood type? Okay. So our antigens are also referred as agglutinogens, because they can also do agglutination, means clotting. Okay. And human beings also typically have approximately 30 naturally occurring antigens that can possibly sit on these RBCs. So presence or absence of this antigen will actually classify. Sometimes not everything, all the antigens will be present in everyone. So if we have something present or something absent, depending on that, we will determine the blood type. Now, the most common antigen is AB, based on which we determine ABO blood group, and the second one is the RH blood group. This is determined by the presence of an antigen called D antigen. So these are the main two blood groups that human beings have, right? Let's first talk about the most major one, ABO blood group. So this means that a person either has an A antigen or a B antigen, or agglutinogen, same thing. So for example, type A will only have A agglutinogen or antigen. Type B blood group will have only B antigen or agglutinogen. Type AB means this person have both type of antigen, A and B. And type O means they don't have anything, nor A nor B. Alright. Now blood will also contain antibody against a foreign blood. For example, a person with A blood will have anti-B antibody, because a person with A blood can only have A blood. It cannot have B blood. So it will have anti-B antibody, because antibody is something that will fight with the foreign substance. Now, a person with A blood group, for this person, B is a foreign stuff. Like having a blood which has B antigen or B blood group is foreign. So a person with A blood group means this person's RBC has A antigen. Therefore, it will not like any B blood group. Therefore, the antibody this person will have is anti-B. Okay. Anti-A or antibody form in blood group around 2 months. It starts forming, and it takes the full adult form around 8 to 10 years. So this is a pretty important chart. So here, suppose a person has A antigen, or that means that person is A blood group. Therefore, they will not like B blood group. So therefore, the antibody this person will have is called anti-B, so that it can reject B blood group if it's given to them. Similarly, a person with B blood group will not like A blood group, so it will have A antibody, anti-A antibody. And a person with AB will have both blood groups, but this person doesn't have any antibody in their body, right? So therefore, since they don't have any antibody, so they can receive blood from anyone. They can receive from A, B, or even O. Why? Because O is exactly the opposite. They don't have any antigen but have both the antibodies. So it's not a bad thing if a person A receives O blood group, because there's no antigen, so they will not clot against each other. Having more antibodies is good, so that's not a big deal. So therefore, a person with A blood group who doesn't have any antibody, only antigen, they can have blood from anyone, so they are called as universal recipient. As opposed to that, O blood group have both antibodies, so they can give the blood, but they cannot have from everyone. So they are called universal donor. So a person with O blood group can only have blood from O, cannot have it from A, B, or AB, but it can give. You will actually do a experiment in lab. Then you basically in hand experiment where you will determine blood groups. Now, the other blood group that is important is called Rh blood group. There's 52 named Rh factors that are present in the human body. Rh plus indicates the presence of D antigen, and 85% of Americans are Rh positive, means they do have this antigen. So this Rh factor or D antigen is basically present in blood groups. So that is, when it is present, then it is called as Rh positive. When it is not present, then Rh negative. Now, if a person is Rh negative, so they don't have this Rh factor or D antigen on their blood cells, so naturally they will not form any anti-Rh antibody, because they don't even have the antigen. So there is no particular antibody against this particular antigen. Okay, and this particular thing is critical if a woman is pregnant. Why is that? So let's talk about that. So suppose woman who is Rh negative is impregnated by a man called Rh positive, and then the fetus is also Rh positive. So what will happen? So when the placenta is formed during first pregnancy, so because when a woman is pregnant, then the placental and the mother's blood is exchanged. Now, since that is exchanged, so the mother will start forming an anti-Rh antibody. Why? She will become sensitized. Why? Because she is now exposed to something foreign. So she is exposed to Rh factor. Previously, she didn't know her body did not know what it is, but now she knows. So therefore, it will be formed, and anti-Rh antibody will be formed. So that is not a problem for the first baby. But if she becomes pregnant for the second time, and the fetus is again Rh positive, then she already has developed those anti-Rh antibody, but the fetus itself is Rh positive. So this anti-Rh antibody can cross the barrier and then can sort of kill the fetus, because anti-Rh antibody is there in the mother's body coming from the first pregnancy. It can cross the barrier, placental barrier, and can attack the fetus blood. Now, can this be prevented? Yes, it can be. For this to be prevented, a RhoGAM serum is given, and this has to be given though within 72 hours of giving birth. So what this product does, this actually prevents an Rh negative mother's antibodies from reaching to the baby's Rh positive red blood cells, so that those antibodies don't reach the fetus anymore. And then in that case, then the fetus or the baby will be protected. So but it has to be given within 72 hours of uh, giving birth. Now this process, if this happens, this situation happens, then this is called as hemolytic disease of newborn. Okay, only occurs, and this will only occur with Rh negative blood group mom and with an Rh positive fetus. If both are positive both are negative, then it will not, nothing will happen. Okay. Now transfusion reaction. What is this? Now transfusion reaction can only happen if there is any blood like mismatch. Suppose a person receives blood from B, then of course it's not going to like, and that is when the transfusion reaction will happen. Donor cells will be attacked by the recipient's antibodies, and therefore blood will start like clotting or clogging the blood vessels, because the blood will start to like clog or clot. Why? Because there will be reduced and the RBCs are not matching with each other, so therefore there will be, the RBCs will be destroyed, there will be diminished oxygen carrying capacity, the blood vessels will be blocked because blood is clotting, and then of course the kidney tubule will fail, so that also leads to renal failure. This actually, the symptoms are like fever, chills, low blood pressure, because basically blood is not functioning, rapid heartbeat, nausea, vomiting. Immediately this blood has to be washed out from that person. Therefore it has to be remembered that again, type O blood group has universal donor, so this can be given to everyone because this doesn't have any antigen. And if a person is AB, then that person can receive blood from A or B or from O, because AB has no antibody in them, right? From this chart, if you remember. So there's something called autologous transfusion, means the patient pre-donates his own blood, and so that it can be used if needed. So suppose this is a type of blood typing experiment you will do in lab. So suppose you have a and like type let's start with A. So suppose you have a person with A blood group, and if you give anti-A antibody, okay, and then what will happen, only the A blood group will clot, because the A obviously does not like the anti-A antibody. Nothing will happen in B, but if you give anti-A antibody and to a blood group and that clots, that means that blood type is A. Okay. Similarly, if you give blood like the anti-B antibody and that blood clots, because that means that is a B blood group, because it doesn't like the anti-B antibody, so that means B. If both clots, that means that is AB. Okay, one is reacting with one another, and nothing clots means that is O. Other than blood typing, other diagnostic tests that typically is done before giving the blood, for example, low hematocrit, we discussed about that. Glucose test, if the person is diabetic or not, leukocytosis. Okay, sometimes like, and then microscopic examination, abnormal size and color or shape of RBCs, for example, sickle cell anemia or like that. So that is also checked before blood is given to another person. Okay, last two concept checks from this chapter, which means which agglutinins are naturally present in someone who is B positive. Okay, so which agglutinin means the antibody is present who is B positive. So let's quickly go here. So B positive blood group, right? So this is the blood group. What kind of antibody is present there? Anti-A. Why? Because it's a B blood group and it does not like A, right? So it's a B blood group, does not like A, so therefore it has to be anti-A. The next one, if John has O positive blood group, he will have to, which of the following will be his antibodies? Now you will remember, again go back to the slide again and remember, and then figure out what antibody he has. Remember, he has O blood group. Okay, so work that out yourself. All right, so these are all the concept check, and which means we have covered now 2 chapters. And so work on this concept checks, and then this will be due by 22nd of January, end of night, 11:59. All right, so make sure you are finishing this up, typing them, and putting it by 22nd of January. All right, see you in the next chapter. Talk to you in the next chapter.